All right, so this is part 2 of chapter 21's lecture. We're starting with the regulation of metabolism, talking about the role of insulin in the, in absorbing your calories, absorbing your nutrients. So this is an overview. Insulin secretion is going to increase during the absorptive state. So when you are getting ready to eat and also while you're eating, the insulin levels will rise, so that your body cells will be in this anabolic state. Insulin is going to help your cells take in energy and build up larger molecules from smaller molecules. So the insulin is going to be secreted when the beta cells of the pancreas detect a rise in blood glucose levels in the blood. Increased amino acid levels in the blood is also going to signal insulin secretion. And then the parasympathetic nervous system. So remember, the parasympathetic nervous system is the rest and digest part of your nervous system. So when you think about food or when you're smelling food, that parasympathetic nervous system can actually send signals to the pancreas and start the secretion of insulin, because it's basically like, get ready, we're going to start eating, food is coming, right? And when insulin goes down during the post-absorptive state, right, it's because your insulin levels have decreased, and it's conversely, your sympathetic nervous system has a role in dampening down insulin secretion, and namely by the hormone epinephrine. All right, so this picture down here is just to show you, remind you the islet, right? This is a pancreatic islet. These are the exocrine glands. Don't worry about those for this lecture. Those are going to secrete digestive enzymes. But then we see our collection of beta cells that secrete insulin and our alpha cells that secrete glucagon. So let's talk about transporting glucose into the cells for just a second. So we know that glucose transporters are on all the cells of the body, and there are 13 of them. They're GLUT1 to GLUT13, but there's 5 that are probably the most predominant ones that we'll talk about. So GLUT1 is going to be on your blood, the blood-brain barrier, and to a lesser extent the heart. The heart has some of this. And notice that it's insulin independent, meaning that these transporters are going to always be present, and they're going to transport glucose into the cells, whether insulin is there or not, right? So this can always allow glucose to go into the cells. GLUT2, you can see, is found on the liver and the pancreas and the small intestine. So this is, again, insulin independent. So if you look, actually, what's interesting here is that the GLUT2 has a very low affinity for glucose, meaning that the transporter doesn't really bind glucose that tightly. And when this happens, the concentration of glucose makes a difference in how much glucose goes into the cells. So the more glucose there is, the more transport you're going to have into those cells. So you actually need a high glucose concentration in the bloodstream in order for a significant amount of glucose to enter into those cells, because a moderate amount of glucose or a low amount of glucose in the bloodstream is not going to be enough to bind to those transporters and to move it into the cell. So if you think about the pancreas, if you think about the beta cells of the pancreas, how do they know if there's blood sugar, right? So how does your pancreas know that you just ate something and then the glucose levels are high in the bloodstream? So they have the GLUT2 transporter, which is only going to transport glucose into the pancreas when it's at very high concentrations. Okay, the liver has the same kind of transporter in small intestine. If you look at GLUT3, this is the brain, your neurons, and sperm. So your nervous system, we talked about how the nervous system only uses glucose for energy. So GLUT3 is, again, insulin independent. You don't need insulin for the brain and neurons to take up glucose. And it has a high affinity. And that should make sense, because you want your central nervous system and your neurons to take in sugar whether or not there's a high concentration or a low concentration. So because these transporters have a high affinity for glucose, they'll take glucose up even at low levels, even if you're starving and there's just a little bit of glucose in your blood. who's going to get that glucose? Your brain, all right, and the nervous system. Then we have our insulin dependent, right? So this is the cells that have the GLUT4 transporter. The GLUT4 transporter has to bind to insulin in order for the GLUT4, well, there's an insulin receptor, right? So there's the insulin receptor, and binding to that insulin receptor will transport the GLUT4 onto the surface of the cell membranes to allow the glucose to come in. So the presence of GLUT4 in high concentrations depends on insulin being present. And the skeletal muscle and adipose in the heart in this way, we're really focusing on skeletal muscle and adipose though. And these guys have a moderate affinity, no big deal, not important to remember, but GLUT4 is this insulin dependent. So your muscles and your fat need insulin in order to be recognize the glucose or take in the glucose through facilitated diffusion. GLUT5, I'm not going to worry about this. It's on your intestinal lining. It's on the luminal side, which means it's the side of the intestines that faces the lumen, right? So you can see the arrows pointing there. The apical surface of the cells will have these GLUT5. Again, independent of glucose or not, it's going to push glucose that's in the lumen, transport it into the cells. It also has a fructose transporter, but again, not important. I really wanted you guys to see how there are glucose transporters that are insulin independent as well as insulin dependent. There are some with low affinities and there are some with high affinities. And it should make sense that the ones with high affinity are going to be the cells that need glucose the most, and these are going to be your nervous system cells. They only use glucose. They're going to need glucose, readily available no matter what, even at low concentrations, so they're going to have a high affinity. And then the pancreas, we're going to get to this in a second, but the pancreas is this is the way it realizes that there's a higher concentration of glucose in your bloodstream that you just ate, because glucose does not enter into the pancreas at low concentrations. You actually need a very high concentration right after you eat in order for the pancreas cells to flood with glucose. So now that we know that, let's take a look at how, we'll come back to this flowchart in just a second. Let's talk about how your beta cells secrete insulin, okay? So glucose, so here we're at step 1. Glucose is in high concentration outside the cell, right? You just ate. It's in high concentration. And it's going to move into the cell via facilitated diffusion with our GLUT2 transporter, which is insulin independent. It has a low affinity, but since there's so much glucose, right? Since there's so much glucose in the plasma, it's going to come in. And so here's our glucose. Glucose is converted to pyruvate, blah, blah, blah. This is the whole cellular respiration pathway. We don't have to talk about this. We're making ATP. So ATP, as you get more glucose, right? More glucose is going to be broken down to ATP. We're going to increase the ATP concentration inside the cell. Now, there is an interesting potassium channel, which is ATP gated or ATP selective potassium channel. So the more ATP there is, it's going to it's going to close. It's going to close our potassium channel. So potassium is going to stay inside the cell. So if we have more positive ions trapped inside the cell, where normally they would leak, right? But since we have this positive charge building up inside the cell, we have a depolarization event. All right, so the potassium movement that's blocked is going to cause depolarization. Now that depolarization is going to open up our voltage-gated calcium channels. Calcium is going to flood into the cells, and that's going to promote exocytosis of our insulin, which was stored inside of our vesicles of the beta cells. Okay, so this last part should look really familiar to you. This is how neurotransmitters were released. It's the same way how insulin is released. All right, but pretty fun way of the pathway of how insulin is going to get secreted. All right, so coming back to this flow chart, okay, so we have an increase of insulin secretion, so beta cells have produced insulin. Insulin is in the bloodstream. And what does insulin do, right? So insulin is going to, for most tissues, is going to increase the glucose uptake, except for the brain, the liver, and exercising muscles. It's going to increase, the insulin is going to also prompt an increase in amino acid uptake. It's going to prompt protein synthesis, and it's going to stop or inhibit protein breakdown, right? So remember, this is an anabolic. We want to build up. So we want to uptake our nutrients into the cells, and we want to synthesize. We want to build protein. In our fat cells, our adipose tissue, we're going to increase our fat synthesis, right? Our fatty acid and triglyceride synthesis, we're going to increase that so we can store our energy as fat. And we're going to stop the breakdown of fat. So we're going to inhibit lipolysis. In our liver and muscle, because they both have glycogen, insulin is going to increase your glycogen synthesis. So it's going to prompt your glucose coming into those cells. It's going to promote those glucose molecules being linked up to make glycogen. And it's going to inhibit your glycogenolysis, right? It's going to stop the breakdown of glycogen. That should make sense. And in the liver, right, we also can make fatty acids. So it's going to promote fatty acids being made in the liver and triglyceride or fat synthesis. And it's going to inhibit gluconeogenesis. So why would you make new glucose from scratch, right, if you're actively absorbing and you're taking in nutrient molecules? So it's going to inhibit gluconeogenesis. So all of that should hopefully make sense to you, that in this absorptive state, right, when you're actively taking in nutrients, insulin is the hormone that's going to promote the massive uptake of these nutrient molecules into the cells, and it's going to promote building up and storing these molecules as fat or as glycogen. All right, so if you're looking at your outline and we're looking at the actions of insulin also, which I didn't talk about yet in the picture, I don't have a picture of it, but it also mediates the transport of glucose into the cells with the GLUT4 transporter. So for adipose, right, it's going to increase. Let's just look at this picture here. Actually, never mind. Let's just stick with the plan. So it's going to promote GLUT4 transporters being put into the plasma membrane, right? So insulin is going to mediate the expression of GLUT4 transporters onto the membranes of cells so that those cells can actually take up more glucose. And what insulin does, if you're looking at little I, 2II, or insulin can actually stimulate transcription and translation of new GLUT4 transporters in these cells. Okay, so insulin is a major hormone for energy storage. It's going to inhibit catabolism, and also it has a permissive relationship with growth hormone. So it's going to promote growth, but the growth hormone is only going to be active if insulin is present. All right, so let's talk about glucagon. So this is just what I was talking about, right? Actions of insulin. It's an anabolic hormone. It's going to build up our energy stores as glycogen as fat. It's going to promote glucose for energy use. It's going to help cells uptake the glucose with GLUT4 transporters. Either, again, I'm just repeating myself, either by putting the transporters up on the cell membrane, right? Or it can actually promote the DNA expression, transcription, and translation of your GLUT4 transporters. It's going to stop catabolism, and then here's our growth hormone permissive relationship. All right, let's look at glucagon. This is an antagonistic hormone to insulin. It does the opposite job of insulin. So it's a peptide hormone, just like insulin is, and it's secreted from alpha cells of the islets of Langerhans. And this is going to be the opposite, right? So whereas insulin promotes anabolism, glucagon is going to promote catabolism, and you're going to start to break down your energy stores. So glucagon is going to be secreted when your glucose levels fall, okay? So Glucagon secretion. It's going to be secreted when the glucose levels drop. And I do have a number on the page for you. Don't worry about memorizing 80 milligrams per deciliter, but your body has a normal range of blood glucose levels. When it falls below that, your glucagon will be secreted, and then it will promote the mobilization of your energy stores, because what's happening is you're starving and your body needs to be able to start up breaking down the energy stores so that you can have energy in your bloodstream for your body cells. Another factor in glucagon secretion is the sympathetic nervous system. So fight or flight response, this is also going to mobilize your energy stores, because glucagon is going to be sort of a hormone to liberate energy stores in the bloodstream. Epinephrine is going to mimic this. Epinephrine also inhibits insulin and then allows glucagon to be released. Secretion decreases during the absorptive state. So when you're eating, you're going to see an increase in glucose in the plasma, and that insulin, that is, we just talked about how insulin gets pushed out, exocytosed out of the cells when there's a high glucose concentration in the blood. Insulin inhibits glucagon. So insulin is actually the sort of dominant hormone here. When insulin's present, it's going to stop glucagon from being secreted. Okay. So here's our flow chart. So alpha cells in the pancreas are going to secrete glucagon. In the liver, it's going to promote all of these processes that's going to put energy into the blood, right? So it's going to promote glycogenolysis, breakdown of glycogen. It's going to stop glycogen synthesis. It's going to promote gluconeogenesis to make more glucose from other molecules. It's going to promote ketone synthesis, because remember, ketones can also serve as an energy source to the central nervous system. It's going to promote peptide breakdown, protein breakdown, and it's going to inhibit protein synthesis, right? So basically, we're just breaking down all of our energy stores. In adipose, we're going to increase our lipolysis, right? Breaking down fat. We are going to inhibit the synthesis of fat, because we want those monomers, we want those fat molecules into the bloodstream for other cells to take up. So this is just a little bit of the negative feedback control of blood glucose levels by insulin and glucagon. Here's just your normal blood glucose level. Do not concern yourself with memorizing the numbers. I want you to know the terms, though, right? So hyperglycemia is a high glucose content in the blood. Emia means in the blood. So hyper means above, gly is for glucose. Hypoglycemia is you have low blood sugar, right? Hypoglycemia. And blood sugar is maintained in this normal level, this normal range, by this yin-yang pull of minute-to-minute relationship between insulin and glucagon. So just take a looking, this is, I put this in your outline as well. When you have an increase in blood glucose levels, right, we're going to start right here. We can see that this is going to promote insulin secretion of our beta cells in the pancreas, right? So this is going to secrete our insulin. And remember, this is because that the GLUT2 transporter, it has a low affinity for glucose, so it can sense when there's a higher amount of glucose in the bloodstream, because it's going to get more glucose inside. ATP is going to promote that whole chain reaction, getting an exocytosis of insulin. The insulin is then going to act on the muscle and adipose tissue to upregulate your glucose transport. So the GLUT4 molecule can be that is on the vesicles, right? It goes to the cell membrane. So it's going to increase the facilitated diffusion of glucose. Or we talked about how we can just get DNA expression to just make more glucose transporters from scratch. So in muscle and adipose, it's going to promote that glucose transport, and the glucose is going to go into the muscle cells and into the fat cells. So then the overall blood levels of glucose will go down because it's going in the muscle cells and inside the fat cells. Insulin on the liver is going to promote glucose storage. So the liver is going to take up more glucose. It's going to store it as glycogen. Remember, the liver can also convert it to fat. And then it can also help in protein synthesis. Okay, and all of those, the fact that the liver is also taking up glucose is going to decrease the blood plasma concentration of glucose. When you have a decrease in blood plasma concentration, that's going to, if you look at this loop, right, it's going to negatively feed back and stop the beta cells from secreting insulin, right? Because again, they're sensitive to the concentration of insulin in the bloodstream. When it drops, it's no longer going to be secreting insulin. Now let's look at glucagon, right? So the alpha cells, so we have a low plasma glucose level. It's going to feed forward. It's going to stimulate our alpha cells to make glucagon. And the glucagon is going to then promote all the catabolic reactions. It's not in this flow chart, but it's going to promote all those catabolic reactions that we just talked about with glucagon. And then we can see here that there is the insulin, it's going to stop. This little symbol here is to stop the production of glucagon. Okay, so that is the flow chart here. And then this is your textbook's flow chart in terms of negative feedback. So this should make a lot of sense at this point. So we can start here with our initial stimulus of high plasma glucose levels. Coming into the beta cells of the pancreas will increase your glucose secretion. In most tissues, it's going to help glucose uptake into the cells by that GLUT4 transporter. Muscle and liver, they're also going to take in more glucose. They're going to promote glycogen synthesis. Okay, so remember, insulin is anabolic, and it's going to shut down glycogenolysis, right? So we're going to build up our glycogen stores in muscle and in the liver. In the liver also, we're going to shut down gluconeogenesis, right? So all these processes are going to decrease the plasma glucose level, which negatively feeds back, right? So we can actually should negatively feed back to here, right? So the insulin levels will go down. Now, if you look at the low plasma glucose levels, the alpha cells in the pancreas are going to start to secrete glucagon. And glucagon in the liver is going to promote catabolism. So it's going to promote gluconeogenesis. It's going to cause the liver to make sugar from scratch, and glycogenolysis, right? Breaking down the glycogen. And that's going to be liberated into the bloodstream, and it's going to increase blood plasma glucose levels. It's also going to promote fat cells to increase the catabolism of fat, lipolysis. And it's going to liberate those free fatty acids into the bloodstream, and it's going to promote the cells to use fat, all the cells of the body to use fat first, and leave the glucose for the central nervous system, right? So that, remember, that's called glucose sparing. So that is going to be another thing that glycogen does. It promotes glucose breakdown, yes, from glycogen, but it also promotes fat breakdown because, again, you want your glucose that's in the bloodstream to go first to the central nervous system, and then your other body cells are able to use fat as an energy source. And then when you when you raise the blood plasma glucose levels, it's going to feed back and stop the production of glycogen. All right, so that is the insulin and glucagon relationship. This is just talking about the effect of epinephrine. And epinephrine, remember, this is adrenaline. The sympathetic nervous system is your fight or flight response. And so when you're creating epinephrine, you're in a state of fight or flight. So you are ready to fight something or you're ready to run away. But either way, you're going to need a lot of energy stores. So your body starts to do the same thing that glucagon does. It liberates energy molecules into the bloodstream so that your cells can use the energy molecules. So it's going to suppress insulin release because we don't want our body to be in the anabolic state. We want our body to be in the catabolic state. It's going to stimulate glucagon release, and it's going to be very characteristic of everything that glucagon does, the post-absorptive state. All right, so for diabetes, Energy metabolism is impaired. There is an energy deficiency for type 1 diabetes, insulin deficiency in type 1 diabetes mellitus. So your body in type 1 diabetes does not make enough insulin, and so therefore your body cells don't take up the glucose, and the glucose remains in the bloodstream. Or you are deficient in the insulin response. So type 2 diabetes is where you have insulin being secreted, but there's some problem downstream. So maybe the target cells are not responding to insulin, or some kind of secondary pathway is not responding to insulin. So that's type 2. And the signs of diabetes mellitus is hyperglycemia, having a lot of blood sugar. Right? So this is a sign that your body, when you eat, the sugar is not being absorbed. There's something wrong with the absorption process, so the blood sugar stays high. And then we have, so normally this suppresses the glucagon release, but it doesn't happen in diabetes. Decreased insulin, right? We have a problem with, there's a decreased amount of insulin, and this decreases the alpha cell's permeability to glucose. It triggers a falsely low glucose level, and the alpha cells increase glucagon to release. So this is where your body thinks it's starving. So because your body thinks it's starving, glucagon's released, and it's going to start that catabolism of your body's energy sources, because there is this broken sort of detection of glucose levels. All right. So let's do a quick review of part 2, glucagon and insulin. Which of the following is not a factor driving increased insulin secretion? You pause it. Okay. So if you're increasing the glucose concentration, if you're increasing the insulin, right, what's going to increase insulin secretion? Yes, glucose is going to increase insulin secretion. Yes, that's good. I didn't actually talk about this one, so let's just ignore that. And then your answer is B. If you increase epinephrine, it actually shuts down insulin. Which of the following is a factor driving decreased glucagon secretion? So what's going to decrease glucagon? All right, it's going to be an increase in plasma glucose, right? So glucagon is stimulated by low glucose. So a high glucose is going to decrease the glucagon. Which of the following liver functions decreases in the presence of glucagon? So glucagon, again, is going to be catabolic hormone. So you want to break things down. Glycogenolysis is yes. This is yes because we want the ketone. Ketones can be used as an energy source for our central nervous system. Protein catabolism, yes and no. So this is your answer. We're not going to build up proteins. All right, and that's the end of part 2.